Hi, I'm Dr. David Seastone. I'm Assistant Professor and Director of Biochemistry at the Lake Erie College of Osteopathic Medicine. In addition, I'm a resident at the Department of Internal Medicine at the Cleveland Clinic in Cleveland, Ohio. It's my pleasure to be with you here today as you embark on this journey in trying to master the material you need to perform well on your USMLE exam. Now, how do you want to perform on the exam? Do you want to perform here, or here? You want to perform up here at the top. So that's where we're trying to get you. I think from my own personal experience with the exam, I can give you some wonderful insight as to how you need to prepare for this and the topics that are going to be critically important that you master prior to taking your exam. For the next several hours, I want you to think of me as your Jedi Master, your biochemistry Jedi Master. I am Yoda, and you're trying to learn biochemistry.
Much information do I have to give you?
Okay, so I'm Biochemistry Yoda. Here we go. So let's start out with Chapter 1, which is going to look at nucleic acid structure and organization. Shown here is the central dogma of molecular biology. Now, you've seen this particular figure, I'm sure, many times throughout your long academic career. The central dogma itself doesn't have that much relevance to what you'll be tested on your exam, but it's an excellent way for us to outline the processes that we will talk about that you will be tested on. So let's start out with the central dogma, and we look at first replication. DNA, as you know, is the hereditary material. It's the blueprints for life. It's what makes one person unique from another person. So DNA has to have an ability to make more copies of itself. In other words, it has to have an ability to replicate itself. So there's a process of replication that we'll be talking about. Also, DNA has to have an ability to transcribe itself into RNA information. You see, DNA, even though it's the blueprints and the hereditary material, we don't want to work with the blueprints. We want to make a copy of the blueprints that we can then work from. Of course, our ultimate goal is to translate the RNA into a protein. And proteins are the workhorse of the cell. It's the proteins that make an hepatocyte different from a neuron in the brain. It's the types of proteins within those particular cell types. So the central dogma states that DNA has an ability to replicate itself. DNA has an ability to transcribe itself into RNA, and then an ability to translate itself into protein sequence. Each individual processes we will review. Notice that RNA can be converted back to DNA. This is a special mechanism that we typically think of retroviruses when we think of reverse transcriptase activity. This is sort of goes against the original central dogma of molecular biology, but it's something we will address as we go along. I want you to think of the term gene expression as a mixture of transcription and translation. Transcription and translation together encompass gene expression. So anytime throughout your studies, when you're looking at gene expression, you should be thinking that transcription and translation both occurred, both processes. So let's compare replication with transcription. Both processes are going to be quite similar to one another, but there are going to be some key differences. The types of nucleic acids that you see being made in replication are going to be different than the nucleic acids you see being made in transcription. Now, they'll be extremely similar to one another, but there's going to be a couple of key structural differences that you're going to be, it's going to be important that you know these differences so that you can perform well on the exam. The lengths of the nucleic acids are going to be different. What do I mean by that? Well, if we place one of your chromosomes end to end on this slide, and we see that it has a certain length associated with it, and we say that that's a DNA. Okay, that's what your chromosomes are. They're made of DNA. Now, if we want to compare the length of RNA to this DNA, is it going to be bigger? Is it going to be the same size? Or is it going to be smaller? How big would that RNA be? It would actually be like a little dot, like a little pixel, compared to the large strand of DNA. So the point being, RNA is much smaller than DNA is. And actually that has some utility for your studies, because when DNA is analyzed in the laboratory, whether it's the clinical laboratory, whether it's the research laboratory, whenever DNA is being analyzed, it has to be cut into smaller pieces to be examined. Can you remember the name of the enzymes that do that cutting mechanism? They're called restriction endonucleases, and they will be reviewed later. Now, the RNA is very small, so anytime you're analyzing RNA, it can be analyzed directly without cutting it into smaller pieces. The enzymes involved between these two processes are going to be different. It's very similar, but also different at the same time. So for replication, you're replicating deoxyribonucleic acid. That's what DNA stands for, deoxyribonucleic acid. And so there's going to be enzymes called DNA polymerases. When you're transcribing DNA into RNA, you're making ribonucleic acid. And so the enzyme that you need are called ribonucleic acid polymerases, or for short, RNA polymerases. The phases of the cell cycle where these processes occur are going to be quite different. For example, transcription and translation, for that matter, are going to happen throughout various phases of the cell cycle, whereas the replication mechanism is specifically going to happen during the S, or synthesis phase of the cell cycle. So here's a picture of the cell cycle. The cell cycle is a topic that you need to be very familiar with for your exam. You might be tested about it from the point of view of biochemistry, from the point of view of histology, from the point of view of pharmacology. So it's well worth your time to understand this cell cycle concept. So how does this work? The cell can live its life in a cyclical fashion. Most cells that aren't very active, they're non-dividing, they're senescent cells, they live their lives primarily in what we say the G0 phase. The G0 phase is a non-dividing cell where you'd find it mostly. Good example of this would be a neuron. The neurons are just hanging out. They're not really actively dividing. Okay, what they're doing is they have a sodium-potassium pump. They're pumping sodium out, they're pumping potassium in, sodium out, potassium in, and they're just doing their thing, not actively dividing. So they're living their life mostly in the G0 phase. Cells in the G0 phase can flip and actually go into the G1 phase, which is where you would find cells that are more actively dividing, more actively turning over. The best example for this would be something like an intestinal epithelial cell. An intestinal epithelial cell is constantly being turned over, new cells are being made, so they don't really hang out in the G0 phase very much. They hang out mostly in the G1, getting prepared for the next phase of the cell cycle, which is the S phase, the synthesis phase. The S phase or synthesis phase is where DNA replication occurs. So later on, when we talk about DNA replication, that's the phase of the cell cycle where it's happening. Notice there's a G2 phase of the cell cycle. This is when ribosomes are dividing, when the mitotic apparatus is sort of getting set up. It's not really true mitosis yet, but the G2 phase is the next phase prior to the cells entering mitosis. That would be your M phase, shown up here, the M phase. So the question is, when does replication happen, and when does transcription, translation, or gene expression, when does that happen? Now you see here that the S phase is primarily where the replication machinery takes place. The transcription and translation machinery, i.e. gene expression, is going to take place throughout all phases of the cell cycle. The G1, the S phase, the G2 phase. But notice something. It doesn't take place during mitosis. So if you're ever asked a question, related to the cell cycle and something about gene expression, there's one phase of the cell cycle that's an exception, and it's mitosis. There's no true gene expression going on there. That's a specialized portion of the cell cycle where the sister chromatins are pulled apart. Sister chromatins are pulled apart, and they then each go into a daughter cell. So that's the cell cycle. So in a typical cell, what's going to get a signal to get the cell to go from the G1 to the S phase? There's going to have to be some sort of signal that tells a cell that's typically just hanging out in the G1 phase to go into the S phase. Well, that signal for our purposes is something like a growth factor. Could be insulin growth factor, could be epidermal growth factor, could be platelet-derived growth factor. Some type of growth factor or hormone is going to tell a cell to go from the G1 phase and to start duplicating its DNA in the S phase. We'll look at many of those growth factors throughout this biochemistry review series. A very high-yield topic that you're going to be expected to know and answer questions about is drugs that target specific areas of the cell cycle. In particular, these are the chemotherapeutic drugs. There's drugs that target the S phase specifically, drugs that target the G2 phase, the G1 phase. There's non-cell cycle specific drugs. There's drugs that target the mitotic phase. So for the S phase, two important drugs that you should remember that target are 5-fluorouracil and methotrexate. Also know the mechanism of action behind these two drugs. Very high yield for your Step 1 board exam. 5-fluorouracil, do you remember what it targets? Well, it's going to block an enzyme called thymidylate synthase. Thymidylate synthase is an important enzyme to convert DUMP to DTMP. We'll review that later, but for now, remember that 5-fluorouracil, which blocks thymidylate synthase, is going to specifically target then the S phase of the cell cycle. What would you use 5-fluorouracil to treat? You're going to use it to treat, of course, cancer, solid tissue cancers, certain ones. You're also going to use it to target, to treat certain dermatologic maladies, actinic keratoses, and so forth. The next drug, methotrexate, you know that it has important utility in treating both cancer and rheumatoid arthritis, and that methotrexate is going to block the S phase of the cell cycle. How? Well, it's going to block dihydrofolate reductase. Dihydrofolate reductase is important in the recycling of folic acid, and you all know that folic acid is highly important for cell growth, particularly during the in utero portion of development. So methotrexate, 5-fluorouracil knows that they block specifically the S phase of the cell cycle. The G2 phase, there's really only one big drug that you have to remember for this, and this is a biggie. It's bleomycin. Bleomycin is a chemotherapeutic agent used oftentimes to treat Hodgkin's lymphoma. And bleomycin, it's very important that you remember that blocks the G2 phase of the cell cycle specifically. And in fact, can you remember some of the manifestations of bleomycin toxicity? This is a biggie. It's pulmonary fibrosis. Okay, so two things to remember about bleomycin. Number one, that it blocks the G2 phase of the cell cycle. But the second is that it causes pulmonary fibrosis. There's actually another drug that causes pulmonary fibrosis, and it's amiodarone, not involved in cell cycle. Okay, mitosis is blocked by paclitaxel, which you know better as Taxol. So that's going to be a microtubule depolymerizer. So Taxol prevents the microtubules from assembling and therefore causes the mitotic spindle to fall apart and blocks mitosis. Other drugs that block mitosis are going to be vincristine and vinblastin. Other commonly used chemotherapeutic drugs target the mitotic spindle and block mitosis. Finally, some non-cell cycle specific drugs. Cyclophosphamide and cisplatin. So cyclophosphamide, very, very important drug for certain leukemias especially. It's going to block the cell cycle non-specifically. So if you're asked a question about cyclophosphamide therapy, don't pick one of the phases of the cell cycle because you're going to get it wrong. It's a non-cell cycle specific. Same thing for platinum-based therapies such as cisplatin. They're going to non-specifically inhibit the cell cycle. Okay, let's now directly compare gene expression with DNA replication. First of all, gene expression, it produces all the proteins that an organism requires. So that's what it does. It produces all the proteins an organism needs to function. Now, if you're renal tissue versus hepatic tissue versus splenic tissue, you're going to make different proteins. So if you look at gene expression in various organs, it's going to be different from one organ to the next. If you look at DNA replication, however, that's going to be the same for all tissues. The gene expression that happens in the kidney versus the liver versus the spleen is going to be different. But the replication mechanisms that happen in those tissues are going to be the same. So DNA replication, which is responsible for duplicating the chromosomes before the cell divides, is going to be the same for all tissues. We already talked about this a little bit, but transcription is making an RNA copy of DNA, and you're making a smaller sized nucleotide on the order of 10,000 up to 100,000 nucleotides in length. So it's a smaller piece of nucleic acid when you're transcribing into RNA. When you're replicating DNA, you're replicating an entire chromosome. Each chromosome is going to be, say about 10 to 100 million base pairs in length. So when you replicate an entire chromosome, well, that's a lot bigger. It's a lot bigger than the RNA. It's 3 times as big, 2 to 3 times as big as the RNA is. And in fact, the entire size of the human genome is what? Well, it's 3 billion base pairs. So 3 billion base pairs divided into 23 homologous sets of chromosomes makes each chromosome about 100 million base pairs in length. Okay? Now, when does this happen in the cell cycle? We already talked about this, but transcription occurs throughout all phases of the cell cycle with one exception. What was that? It's mitosis. Okay? All phases of the cell cycle. We call those the interphase. Interphase is basically all the phases of the cell cycle with the exception of mitosis. DNA replication is going to specifically happen during the S phase. During the synthesis phase is where you see this going on. Finally, where in the cell does this happen? Because a very easy question to ask you is to give you an electron micrograph, point to part of the cell, one of the organelles or portions of the cell, and ask something about what mechanism is occurring in that particular location. Well, for translation, if you're talking about a eukaryotic cell, it's going to happen out in the cytoplasm with the ribosomes. Whereas for a prokaryotic cell, it's also going to happen out in cytoplasm. Now, if you're looking at a eukaryotic cell for replication, the replication is going to happen within that membrane-defined nucleus. Whereas for a prokaryotes don't have a membrane-defined nucleus, so the replication machinery you could think of as just happening in the cytoplasm as well. But for eukaryotic cells, replication happens in the nucleus, gene expression happens out in the cytoplasm. For prokaryotes, there's not so much of a location difference for the two processes.